So here's the setup, not to scale of course. An oligoaniline is trapped between a scanning tunneling microscope probe, insulated so it can work in an acid environment, and a gold substrate, and break junction measurements are performed. The electrolyte is under the control of a 3-electrode control circuit, such as that discussed when we described how to control the polarization at a surface, so that there are two electric fields in this problem. One is the local field applied between the tip and the substrate, in other words, the bias that drives the current through the molecule. And the second is the polarization that accumulates at this surface when the whole electrochemical cell is polarized by this counter electrode here, with a polarization that is set relative to a reference electrode here. So on this plot is shown, first of all, a cyclic voltammogram for a monolayer of these oligoaniline molecules. And you see as the potential is increased, this first oxidation process here, this peak would represent the formal potential. As one continues the oxidation, the current falls as fewer molecules remain to be oxidized. The current rises again as one reaches this second oxidation of the oligoaniline. Then on returning the sweep, you'll notice the second oxidation is not reversible, but the first oxidation is. So experiments are confined to this region here, containing just this first reversible oxidation. These points here show the measured single molecule conductance, averaged over many thousand measurements. And you see that as one goes from the insulating form here towards the oxidized form of the molecule here, the single molecule conductance rises, and then as you approach the second oxidation here, back into the insulating form of the molecule, the single molecule conductance falls again. So here is a plot of a different experiment. In the last experiment, the probing potential between the tip and the surface was kept very small, as the electrochemical potential of the surface was swept to oxidize the molecule through those two oxidation steps. In this experiment, we now set the potential of the surface and sweep the tip substrate potential. Now, before I show you the results for that experiment, let me show you what happens in a solvent containing no ions, an organic solvent. Because the solvent cannot sustain the polarization needed necessary to allow the oxidation of the molecule, no electrochemical process occurs, and the current versus voltage response of the molecule is simply like Ohm's law. It's linear. But if we now repeat this experiment in acid, so now we are keeping the surface potential fixed and sweeping the tip to substrate bias, a very interesting thing happens. We poise the potential in the state where the molecule was in its first oxidation state and therefore highly conductive. So current rises rapidly as a function of the bias applied between the tip and the substrate. However, when we achieve a certain critical bias, you'll see that the current actually falls with increasing tip bias. This phenomenon is called negative differential resistance. What is happening here is the electric field between the tip and the substrate opposes the surface polarization set by the electrochemical control of the surface polarization. At this point here, the molecules are driven back into their insulating state, and so the conductance falls. This explanation is verified quantitatively by using the data for the variation of the conductance as a function of potential shown on the previous slide and using it to fit these data for conductance versus tip substrate bias at a fixed surface potential. Another example of pH control, in this case of devices, is shown by this beautiful work out of the Knuckles group at Columbia University. Rather than use a scanning tunneling microscope, this group has developed techniques for covalently bonding single molecules into very small breaks between metallic carbon nanotube electrodes. So they used these techniques to place an oligoaniline between the electrodes and then used pH control to switch the molecule between its unprotonated and protonated form. The result is a switching of the conductance, and this plot here shows the current through the carbon nanotube electrodes as the pH is cycled between pH 3 and pH 11, resulting in a high, low, high, low, high, low cycling of the conductance, showing how a single molecule can be used to sense the local pH. So this brings me to the final topic in this unit, which is the use of single molecule methods as chemical sensors. It should be clear from the foregoing discussion that a critical element in single molecule measurements is the connection between the electrode and the molecule. Now, I've talked about covalent connections, but the connections could just as well be hydrogen bonds. If more than one hydrogen bond is used, they have the property, as outlined in Chapter 6, of acting as exquisite chemical recognition elements, because the pattern of donors and acceptors on one side of a hydrogen bonded junction has to be perfectly complementary to the pattern of donors and acceptors on the other side of the hydrogen bonded junction. Thus, if you use molecules that hydrogen bond connected to electrodes to recognize molecules that go into a tunneling gap, one can use chemical recognition combined with tunneling signals to recognize single molecules. A particular example of this is uses of these methods to sequence DNA by generating a unique tunneling current for each of the 4 bases. The final slide shows the use of a chemically functionized tunnel junction. So here is a molecule with a hydrogen bond donor and a hydrogen bond acceptor, the same molecule on a surface, recognizing a DNA base as it diffuses through the tunnel junction. Now, it turns out that for a particular value of this tunnel gap, there is a unique hydrogen bonding pattern that can be formed between these reagents and a DNA base trapped in the junction. So here you see thymine, guanine, cytosine, and adenine. And the result is that for each of these junctions, one gets a characteristic tunnel current, shown here by these 3 shaded boxes, for each of the 4 bases: thymine, guanine, cytosine, and adenine. And therefore, by reading currents from this junction, one can read the sequence of a DNA molecule trapped between these electrodes. This is one area where single molecule electronics might be employed in the very near future for its unique ability to identify single molecules.